This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 307, recorded on March 21st, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from St. Louis, Missouri, Petra Levin.
Hello, it's great to be here. We have, 50 degrees and overcast. Actually, though, the sun just started peeking through.
It's like 5 degrees C here in New York. It was -1 this morning. I don't know what's going on.
We had 25 degrees 2 nights ago, so.
Some cold front went through. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. I know where the cold front is.
Where?
It's here. Here in Michigan.
It's cold there?
Yeah, but only for a couple days. We're going to be 50 by Monday.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. And we have Chamber of Commerce weather, so please come visit. It's 75 degrees, sunny, and the co-eds are out, eating on the lawn.
Wow. What time did you want us to come?
Anytime. You're always welcome, Michele.
I'll be there. I don't think I've ever been to Charleston, except the airport.
The food is fantastic.
Is it?
Yeah.
Yep.
Well, I was in Austin last week, where the food is okay. Everybody makes a big deal out of Austin. Anyway, I was at South by Southwest. This is relevant to
cool.
today. I was asked to chair a session, which was called From Adversary to Ally, Reimagining Viruses as Medicines.
Mm-hmm.
Interesting.
So the recording is available. I will put a link in the show notes. My guests were Stephanie Strath-D, who should be known to TWiM listeners. Her husband, she saved her husband with phage therapy. Well, she didn't. But
right.
She told the physician, "Can you try this?" Because he had a multi-drug resistant Acinetobacter infection that he got in Egypt, and nothing could save him. And she said, "How about phage therapy?" So Ry Young at Texas A&M got a phage that matched his bacterium, and they gave him every day intravenous, like 10 to the 11th phages or something.
Wow.
For 2 weeks, and he woke up and he's fine now.
There's an excellent book called The Perfect Predator, which she wrote.
She wrote
about this whole story. It's actually very gripping.
Yeah. So she, and they're making a movie out of it now.
Oh.
That's awesome.
So she was one of my guests. And the other guest, Andrew Scharenberg, he is the head of a small biotech called Umoja Biopharma. He's trying to redesign CAR T-cell therapy so you don't have to take the T-cells out and infect them. Anyway, they were both good, so you should listen to it. It's just about an hour in length. That was a lot of fun. And that's a good lead-in for today. We have a snippet in a paper, and our snippet is from Michael, which is related to Stephanie Strath-D's story, right, Michael?
Absolutely. Vincent reached out to me before he interviewed, asked me for some questions, knowing that I had previously worked on phage therapy. And so I stumbled across this paper in MBio. It's entitled Bacteriophage and Antibiotic Combination Therapy for Recurrent Enterococcus faecium Bacteremia. And it's a really good science study of what this woman went through and getting her husband cared for. And here in this paper in MBio, you can see with this group of investigators from the University of Pittsburgh's Department of Medicine, Division of Infectious Diseases, as well as Department of Immunology and Microbiology from the University of Colorado School of Medicine. And the authors are Stelfox, Fernandez, Shields, Hadar, Hughes, Kramer, Dubinsky, Manghalea, Aria, Canfield, Derkop, Van Tyne. And this is an absolutely fascinating story. So I'm going to focus on the microbiology of this. And towards the end, we'll get into the specifics of what the workers had to do in order to address the infection in this study. So the microbe in this study is not Acinetobacter like the other story that Vincent just introduced is actually Enterococcus faecium, which is a gram-positive coccus that is a routine member of the human GI tract. But often when this creature leaves our gut, it can result in tragic consequences where it can cause invasive infections, especially in immunocompromised hosts. Additionally, the enterococci display intrinsic resistance to many antibiotics, where in fact most of the clinical isolates of Enterococcus faecium have acquired vancomycin resistance, which in the jargon of hospital infection control professionals is commonly referred to or as a cuss word, which is VRE. Further, microbes like VRE are often resistant to many of the other drugs used empirically before our clinical teams can get the results back from the lab, where they will then know the culprit or the culprits affecting their patients, leaving clinicians with a limited repertoire of effective antibiotics from which they can draw from. And at the same time, because of the empiric prescribing of antimicrobials, this then exposes the microbial community in those undiagnosed hosts, if you will, to an opportunity for natural selection to take place, which results in the development of resistance, which has driven our problem with increasing multi-drug resistant microbes in the patients that our clinicians are seeing. Candidly, this paper made it to TWiM as it well illustrates the challenges facing our medical community as they attempt to purge these increasingly refractory microbes responsible for recurrent infections in patients. And here, because they're recurrent and refractory infections, refractory in this case means they're resistant to many of the antibiotics that we have available, or the patient won't tolerate the antimicrobials. So what they try to do is they try to introduce phage in combination with the antimicrobials to assist the patient's immune system in their ability to clear the infection, or if they can't clear it, at least improve their quality of life. And that's what the paper is all about. And it teaches many important lessons of this tour de force that goes on between clinical medicine, the research microbiologists, and how we're trying to come up with new schemes to care for those with microbial infections. Now, the experiments or treatment plan that the authors developed for this study was simply to assist the patient with their infection, and the techniques are quite familiar to routine listeners of TWiM. It's whole genome sequencing, characterization of the microbiome using targeted metagenomic 16S sequencing, as well as Rich Condit's favorite experiment, a plaque assay, where the team assessed the ability of the treatment phage to cause a lytic or lethal infection of the offending microbe. And then for good measure, the authors provide to us a bit of immunology where they then assessed whether this immunocompromised patient could mount an immune response to the phage, thereby limiting the effectiveness of the antimicrobial phage cocktail through the process of routine immune inactivation. Now, phage therapy, as Vincent introduced us, is often considered an antimicrobial approach of last resort. You sort of need a concerned family member to literally get down on their hands and knees and beg the clinical team to try this. And as a consequence, this therapy often takes some time to put into place simply because of all the checks and balances that need to take place prior to the administration of phage therapy to treat the infection, to ensure that the treatment by itself doesn't harm the patient. Yet, even in spite of these hurdles facing our clinical teams, phage therapy in combination with traditional antibiotics is now an emerging treatment scheme as a possible therapeutic approach for treating these bacterial infections that fail successive rounds of traditional therapies. Now, the reason people are so enamored with phage therapy is it's elegant in its simplicity. You administer approximately 1 billion phage per dose to an infected individual using a bacteriophage, specific to the microbe you are trying to eliminate. In this case, they are using Phi9184, which is a double-stranded DNA non-enveloped icosahedral head-to-tail geometry type virus of about 60 nanometers in size. This has the ability to plaque or manifest itself upon absorption into a lethal or lytic infection in the microbe responsible for the infection. In some cases, a phage combination antibiotic therapy, a cocktail or a spectrum of phages is often used that will plaque many different strains of the offending microbes in order to address the aphorism attributed to Friedrich Nietzsche, what doesn't kill you makes you stronger, where if the phage or the cocktail of phages fails to get them all, the resistant microbe will take over and literally you're back to square one and you have a recurrent infection, especially if the microbe is carrying a robust resistome. And recall that a resistome is nothing more than genetic traits that the bacteria carry that can limit the effectiveness of our antimicrobials through their inactivation or making a pump or some other mechanism.
So, Michael, just to clarify, the cocktail has a bunch of different kind of phage that recognize different parts of the same bacterial pathogen that you're targeting.
Sometimes, yes.
Could be, or is it just they're trying to guess what the pathogen is, so they try a cocktail? They don't always match these perfectly, right?
Correct. The answer to your question, Petra, is yes to both. The Tbilisi group uses a broad spectrum cocktail that works on many different microbes. Here, they were very specific. They literally built their phage.
Right.
The Phi91 and the Phi-3 are specific for this particular strain of Enterococcus faecium. They literally went out and built it.
Right.
And
so this is a very interesting thing about phage therapy, right? I think
yes.
that it's worth discussing because, often when you have a bacterial infection, you don't have time
yes.
to do this kind of personalized medicine, which you can do with cancers or things like that. And so it's a very interesting balance between this sort of tailored cocktail. And I just want to make sure everybody understands.
Yes, and that's good.
It's not like you can go to the hospital and get a phage here.
I need this specific one. No, you don't get that.
That's what the Tbilisi group sort of whole thing, and they've been around for a very long time.
It's interesting. Stephanie Strathy was talking about this. She said there are collections that you can draw from now. There are extensive collections that should be present and can cover a lot of isolates. Yeah.
Great.
So let's set the stage here. This group used a combination approach of two very specific phage that will target the recurrent infection. And it was because this single patient had the same recurrent infection over and over. And it was a bloodstream infection, which is often fatal. And she was having a decay in her quality of life, and it really impacted. So they designed this treatment scheme in concert with systemic antibiotics to treat this single patient with this recurrent Enterococcus faecium bloodstream infection that were up to that point in time non-responsive to the antibiotics alone. And they simply asked a very specific outcome-driven question, whether or not this new treatment approach would result in fewer hospitalizations and improvements to the patient's quality of life. Now, how they did this is beautifully described in their first figure. It's a busy figure, but since it's an MBio open access paper, you'll be able to see it. But it's really, even though it looks busy, it's not. It's really pretty straightforward. Now, the study was done between June of 2020 through July of 2021. And as many of us will recall, it was the scary time where COVID was raging and we were losing about 25,000 people a week to COVID in the United States. So in that first figure, you have two panels. In panel A, you have 3 bars above the timeline that depict the type and duration of treatments used throughout the observation period. The blue line is the Phi-9184, the first phage that was designed to target the Enterococcus. And then you have the red bar, which is Phi-HI3. And they sort of were worried about the patient's immune system making an antibody to take out or lower the dose of 9184. So they have a period where they up the dose of 9184 to 2 billion phage per dose. And then they use a combination of Phi-9184 and Phi-HI3. And then the antibiotics they use, and they changed those up too. They started with daptomycin, and daptomycin is one of these very expensive drugs, which, as an aside, targets the bacterial cell membrane. So which results in the rapid depolarization and death of the microbe. So you have a double death mechanism. You got the phage killing it, and you have it attacking the membrane. So it's on a bar by itself. And then the bar below, you see a mixture of antibiotics they use that are targeting different cellular targets. They, again, use vancomycin, which is a cell wall drug. They use ceftaroline, which is another cell wall antimicrobial. They go to ortavancin, which is another drug. And so they have this myriad of drugs that they are trying to use to effectively sterilize the infection out of the patient, but it has this nasty habit of just keep coming back. And so the bar graph goes into the beautiful molecular biology that they have done on this particular patient, showing the isolations, showing whether or not the VAN-A operon is present or not. They're asking the question, can they delete the bad actor from the patient? And unfortunately, in the end, the microbes adapted to both the phage and the antibiotics, and the patient succumbed to an infection. But before the patient died, they learned that the combination of phage and antibiotic therapy actually did improve the patient's quality of life. They were able to go without IV antibiotics and just do it treatment-wise, taking them in pill form. And
At home.
At home, yes. Please, thank you, Michele. Mm-hmm. At home. But unfortunately, they were never able to purge the bad actor. So they thought that there may have been an antiphage antibody response, and that's what effectively is beautifully shown in Figure 2, where the authors speculated that the patient was indeed making neutralizing phage antibodies. They looked in normal sera from not this patient, but just from normal human sera to ask if, there were inherent neutralizing antibodies. And they literally showed that it was the patient making the antibodies against the phage that was reducing the efficacy. And that's beautifully shown in Figure 2, Panel B. And you can literally see the efficiency of plating fall off a cliff. And
that's kind of an expected result, right? You put a foreign entity in your body, and our body then is very good at recognizing it and making specific antibody to take it out.
Yeah. So summing things up, this study, albeit using an N of 1, well illustrates the potential of this customized, personalized medicine approach as an additional option in the antimicrobial toolbox for treating invasive, at least in this case, enterococcal infections, at the same time highlighting our need for the development of a better understanding of why infections persist and why infections can reoccur. So what do I mean by this? Let's consider the author's top-line conclusion. The combination worked until it didn't, where the partial attribute was likely the patient's reoccurring bacteremia was from the reseeding of herself from her GI tract's microbiota. But consider the challenge that faced this clinical team and how, as microbiologists, we might help contribute to improve on this therapy to assist our clinical colleagues in appreciating the unique microbial environment created by this unique patient. Now, up to this point in time, I haven't described the individual that was cared for. And so I'm going to do that now because I think it will give us, the microbiologists, a sense of the challenge facing the microbiology. Consider that this individual in this study was 57 years old. She was a female with a past medical history significant for a Roux-en-Y gastric bypass surgery. Roux-en-Y gastric bypass is a type of weight loss surgery commonly referred to in the jargon as bariatric surgery. It's often done laparoscopically, where small incisions are placed in the abdomen, where the surgeon simply alters the size of the patient's upper stomach from its normal volume of 1.5 liters to the size of an egg. To the size of an egg. You go from 1.5 liters down to an egg. Now, it's accomplished by stapling off the upper section of the stomach. This then reduces the amount of food the patient can eat and sets the type of substrates being further deposited downstream in the small intestine. Now, the reason it's called Roux-en-Y is because of where the surgeon connects the shrunken stomach to. It's further downstream in the small intestine. So this then reduces the substrates and the enzyme activity because there are specific enzymes synthesized based on the location in the small intestine. Plus, we lose all that neuroendocrine communication between the cecal duodenal junction and the stomach duodenal junction. All of that is going awry. More importantly, because the substrates in the small intestine have changed, the substrates available or the menu of availability to our most complex microbial niche, our colon, where our microbial diversity is at its greatest, is effectively changed, forcing me to wonder if the physical condition created by medicine was partially responsible for this poor patient's continuous recurrence, because we so fundamentally changed the microbiome dynamics that we were unable to, even with our best top-line antimicrobials and the personalized medicine of phage, able to address the reoccurrence of this infection. So I think we have to begin to understand the importance of our whole digestive system in bringing things together.
Michael, is bariatric surgery associated with these kinds of infections previously? Because a lot of them are done.
A lot of them are done. I don't know. It wasn't mentioned and I didn't have time.
Yeah.
The other problem is this patient has also been suffering from recurrent extended spectrum beta-lactamase producing gram-negative urinary tract infections and pulmonary infections. So again, her microbiome is harboring resistance genes against many of the first-line antibiotics. Wow. Additionally, the poor woman was also suffering from Sjogren's syndrome, which is an autoimmune condition that often occurs with other ailments such as rheumatoid arthritis and lupus. And if that were not enough complexity for the microbial community in her gut to consider, she was also suffering from an adrenal insufficiency where her body was not producing a sufficient volume of corticosteroids, which have all sorts of implications, especially given what we're learning about long COVID in the role of corticosteroids in the paradigm of clearing infections. So all of these things and more come into play. So we as the microbiologists need to help our clinical colleagues not only understand the clinical microbiology, but they also need to understand that our digestive microbiome is a complex community, and we really need to understand the circumstances and the microbial dynamics around these recurrent, in this particular case, Enterococcus faecium infections.
Michael, thank you for describing her own vulnerabilities. And I really admire the woman and her family for leaning in and wanting to try this therapy. And they point out in their acknowledgments, the authors, that they express their deepest thanks to the patient and her family. Without their assistance and perseverance, we would not have been able to share our findings with the scientific and medical community. And it was the patient's sincere hope that her experience would inform further studies to help future patients with difficult enterococcal infections. So thank you.
It's a powerful story, and it should give every young microbiologist pause, because this is what we need to be thinking about: recurrent infections and this whole microbial dynamic that is taking place simply because of the intervention of a surgeon.
And it was just one person, but I have to say I was really excited and hopeful when I saw how what a great job those two phage did about knocking back her infection and improving her quality of life for a number of weeks.
Yeah, this study went on for well over a year. So she did get better. She then got worse. She then get better. So
that's the problem when you have extended infection and replicate. You're going to get resistance when it's so long. I think the Stephanie Strathy story was much more acute. And
yes,
right. Yeah, so, here they didn't see resistance, but rather, the body raising the antibody, which makes sense. Clear out the phage that are doing the work.
But also these extended infections, I think for the same reason antibiotics sometimes are challenging. It's not only resistance, it's also where the bacteria are, and you get most of them, but not all of them. And then even with the phage, that's very difficult.
So in the end, Michael, the phage, the isolates were still susceptible to the phage?
The isolates are still susceptible.
But it was her antibody response.
Yes.
So that's where we need a cocktail of phages with different antigenic sites that you could immediately switch to, right?
Yep.
Yeah, not a cocktail, like a series.
Yeah. Not a mix, because you want to give them one after another, right, as they change. Yeah. But that requires some research and investment.
Right.
And the challenge, this is what got me out of the phage therapy business, is the FDA. It's because it's so expensive to take each one of the developed products through the pipeline. It's just financially untenable.
I mean, it's interesting, though, because like WashU has a big personalized medicine cancer center, and obviously people are putting a lot of money into that. And that had, I guess, once you get to the CAR T cells, the FDA is not as, you're sort of repeating, wash, rinse, repeat, and here you're putting in different phage. But also the thing is, like, what is the numbers that we're dealing with these very specific chronic infections? Like what, financially for the institutions or the NIH, how much, this is always the problem with infectious disease, right? It's not the money in it for antibiotics or any antimicrobial is not necessarily worth the R&D. And
they're not high blood pressure pills.
Yeah, and they're not even cancer medications. So, yeah. They're not old people drugs.
Watch it now.
I know, I'm old.
We all resemble that remark.
That's why I feel comfortable saying it, Michael. So.
Thank you, Michael.
Thank you.
Now, Michele, hopefully you have a little more uplifting story for us.
I think it is, and I think it's also just kind of weird and wonderful. The title is Dopamine Receptor D2 Confers Colonization Resistance via Microbial Metabolites. And it is published in Nature. It's now available online. And this was work from Samantha Scott, Xingjing Fu, and Pamela Shang, who are at Cornell University in Ithaca, New York, from microbiology, chemistry and chemical biology, their Center for Immunology, and their Institute of Host Microbe Interactions and Disease. So let's break this down. The first word in the title is dopamine. So you may recall this is a neurotransmitter or a chemical messenger that equips our brains to communicate with a variety of other organs and tissues. And in particular, dopamine is kind of a generalist. It affects many different body functions, including memory, movement, motivation, mood, attention. Here we're going to find out it's contributing to our innate immune system. So colonization resistance is also in the title. What is that? So in addition to our body's standard defenses that you may think about, our white blood cells, tissue barriers, the microbial communities that live in us and on us also can ward off invaders. So a classic example was when we saw that using oral antibiotics increased people's risk of infection in the gut by Clostridium difficile. So what we've learned from studying this and others is that, in its simplest form, in our gut are communities of microbes that form these stable food chains. And they don't leave many crumbs left over for newcomers to come and eat, or room for the newcomers to stick. So that's one kind of natural barrier we can thank our microbes for. But here, Scott and her colleagues make this discovery that microbes feeding in the intestine also release certain metabolites that, in this case, quiet the intestinal epithelium and interrupt this evil genius strategy that certain pathogens use to bind tightly and cause severe bloody diarrhea. So let's talk a little bit about bloody diarrhea before we get into the science. Some of you may recall the dreaded 0157H7 E. coli. It, about 30 years ago, caused this massive outbreak of bloody diarrhea, more than 700 cases across 4 different states, 100 hospitalizations, death of 4 children, all traced to the same fast food hamburger chain. Uh-oh, this is why we're not supposed to eat pink hamburgers. And I will include in the show notes, CDC keeps a track of what outbreak investigations are going on. And right now there's one for E. coli that is traced to raw cheddar cheese. So cheese that hasn't been raised to temperatures that could kill bacteria in the process. So what distinguishes these nasty 0157 bacteria from the garden variety E. coli that live in our gut? For one, they acquired from a phage a gene for Shiga toxin. So this is a protein that can cause tissue damage by blocking host protein synthesis. Some of the strains also acquired, again by horizontal gene transfer, a large block of genes that equip the bacteria to bind to the epithelial cells and then trigger a dramatic movement of the cytoskeleton to erect a pedestal. And from on this stable perch, the E. coli can then deliver their Shiga toxin into the host cell, cause the bloody damage, and in some cases it can progress to kidney damage, which is called hemolytic uremic syndrome. So this is a really beautiful evil genius strategy that has been studied for a number of years, and I'll include in the show notes an animation made by our colleague Brett Finley, who's been studying this for a number of years. It's a lovely animation that shows the steps of the binding of the E. coli and the rearrangement of the actin cytoskeleton to form this pedestal. So here we're going to find that microbial communities in our gut can also protect us, not only from just by consuming nutrients or blocking space, but also some of the metabolites that they release into the gut can short circuit that actin rearrangement that is enteropathogens use to form pedestals. So it protects us from these invaders. So this was a new idea to me, but the Chang lab has been studying this for a while. Pamela Chang published in 2020 a review, Chemical Mechanisms of Colonization Resistance by Gut Microbial Metabolome, where she reviews the literature and shows that a number of metabolites of the amino acid tryptophan can modulate the human host immune response, but they can also trigger a number of other responses in the microbes themselves. And these range from the yeast Candida albicans to different bacteria like Pseudomonas, Vibrio, enterohemorrhagic E. coli. So it's something her lab has been thinking about. And here they make great progress. So in the first experiment, so their overall goal here is to try to better understand why tryptophan appears to confer protection against some infections. So the first experiment was a straightforward one. And it's outlined in Figure 1A, the schematic. They first fed mice antibiotics to clear out their gut microbiota, and then they fed them chow that either contained or didn't contain a supplement of tryptophan. Then after a week, they infected them with Citrobacter rodentium, which is a mouse pathogen that does the same magic trick of forming these pedestals in the mouse gut epithelium to form this stable perch. So if we look in panel Figure 1, panel F, on the far left, we can see what the histology of the gut epithelium looks like normally, beautiful brush border. And then if we look at the third panel, that's the example of what happens after exposure to this pathogen, Citrobacter. And what you can see is that the gut epithelium is extended, the villi are extended and inflamed. And then the very next panel, number 4 in this, if we compare if the mouse had been given a diet that contained a tryptophan supplement, the microvilli looked normal, more like the control panel. So clearly tryptophan was having a direct effect on the pathology, and they demonstrated that it was actually linked to an effect of the microbes themselves, because if they first cleared out the Citrobacter by including an antibiotic, tryptophan was no longer protective. It still causes inflammation. So they quantify these beautiful histology photos in a painstaking series of assays, and those bar graphs you can look at in figure 1C. We see that not only is the pathology protected, the histology rather is protected in the presence of tryptophan, but also the bacterial load is reduced. And then they take careful measurements of the histology and also the this pathology of the microvilli, and that's all shown quantitatively. So now that they have a way to measure this impact of tryptophan, they're curious whether it's tryptophan itself or one of its metabolites. So what they did is collect the feces of the mice that were fed tryptophan or not, and they did mass spectroscopy to look at what the metabolites were. And they actually found 4 different derivatives of tryptophan that correlated with this protective effect. And you can look at the molecular structures. They show those schematically in figure 4J. So to figure out which one, if any, or all of these products were protective, they repeated their same experiment, but this time they supplemented the mouse diet either with tryptophan or one of these particular metabolites, and then did the same experiment. And what they found in figure 2 is much like TRIP, each of the 4 metabolic products of tryptophan also decreased the load of the Citrobacter pathogen and also decreased the tissue damage. So they also wanted to know whether this was an effect on the Citrobacter itself, or was it just changing indirectly the microbial community. So they did 16S profiling of the microbial community and found that that was unaffected by the tryptophan. So somehow this tryptophan supplement was having a specific effect on the Citrobacter pathogen. So they tested the simple model. Maybe tryptophan or its metabolic derivatives were toxic directly to Citrobacter. So they simply cultured the bacterium in broth that either contained or didn't the TRIP or one of its 4 metabolites, and nothing happened. They grew just as well, nothing different with or without these metabolites. So something more interesting is going on, something more complicated is happening in the mouse gut that's protecting the mice from this pathogen. But what? So you can imagine this is a pretty complicated scenario. What they did is first take these the structures, known structures of these tryptophan derivatives, and they used computational prediction tools to just ask, like, what is out there in nature that could bind to these particular structures. And what their computer told them was dopamine receptors. So I'm sure as a microbiologist thinking about host pathogen interactions, a neurotransmitter would not have been on my short list, but there it was. So again, this is a common neurotransmitter, equips our brains to communicate with a variety of organs, but the biology so far is telling them that's what's important. So they tested this directly. Fortunately, they were able to go to the shelf and take advantage of known drugs that can inhibit the activity of certain of these dopamine receptors, and they're called DRD2, 3, and 4. And sure enough, the protection that they'd observed when they fed the mice tryptophan or its metabolites did require the activity of these dopamine receptors. And again, they did this by these painstaking assays of looking at the mouse tissue, quantifying pedestal formation, quantifying bacterial load, etc. But they didn't stop there. They also found that the TRIP metabolites could bind directly to dopamine receptor DRD2. They made epithelial cells that lacked this receptor and found that they were no longer protected from the Citrobacter. They also found that they could substitute dopamine for their tryptophan in the diet and found that, yep, that also protected the mouse epithelium from the Citrobacter pathogenesis. And I really admire these experimentalists. They tested everything. They also engineered mice that lacked the DRD2 dopamine receptor on their intestinal epithelium. and then infected those with Citrobacter with or without the tryptophan supplement. And if they didn't have the DRD2 dopamine receptor, the dietary supplementation with tryptophan or its metabolites did no good. So they're pretty confident they're on the right track, but still they're thinking this dopamine receptor is on other cells, not just the intestinal epithelium. In fact, it's on a number of white blood cells. And to me as a microbiologist, that makes more sense to me. So again, they did the rigorous experiment, and again, they're taking advantage of the Crelox recombination system. And I just want to give a shout out. This is another gift to us in the research community from bacteriophage. It allows us to go in and make specific deletions of particular genes in mice or whatever system you're working in. So they generated mice that lack the dopamine receptor DRD2 in particular white blood cells, macrophages, or dendritic cells, or CD4 T cells. And then using each of these mice, they then did the TRIP supplementation or not, infected with Citrobacter, and sure enough, they found that it made no difference if the macrophages, dendritic cells, and CD4 T cells had DRD2 or not. So they were correct that it is really the epithelial cell sensing the tryptophan metabolites through their dopamine receptor that is conferring protection. So this is amazing. It's weird and wonderful.
So beautiful.
So now that they're confident that they know what the key players are, they were able to turn to the literature where there's been a lot of work studying this fascinating interaction between enteropathogenic E. coli and epithelial cells to form these pedestals from which the bacteria release their toxin and do their damage. And they've shown this schematically in figure 4J, a schematic that shows all of the components. So shout out again to Samantha Scott, who systematically then took advantage of reagents that were available and also antibodies in the literature to test whether each of the components previously identified in this signal transduction pathway were in fact contributing to this protection via tryptophan metabolites. And for example, one of the key proteins known is a host protein called NWASP, which is a key regulator of actin polymerization. So when NWASP is activated, actin then gets polymerized to form these long fibers, and that's what pushes the epithelial membrane up and forms these pedestals that the bacteria sit on. It's also known that to shut off actin polymerization, NWASP gets phosphorylated, and then that targets it for degradation. So now it's no longer firing and triggering actin polymerization, so the cell quiets down, and you wouldn't have this rearrangement. So what they found is, in fact, when dopamine receptor, DRD2, on the epithelial cells was allowed to bind to the tryptophan metabolites, the NWASP protein did get degraded, and that did then short-circuit this actin polymerization that was required for formation of these magnificent pedestals where EHEC and Citrobacter perch and release their toxin and damage the host tissues and cause bloody diarrhea. So I have to say that I have not described in detail every very rigorous, logical, quantitative experiment that they did to rule out, to show the specificity of their assays, to rule out other interpretations. I mean, there is an impressive amount of data in this paper, but it really beautifully shows that when feeding on tryptophan, bacteria release metabolites that can activate receptors on the epithelial cells in our gut that normally bind the neurotransmitter dopamine. But a consequence is that the host then loses a key regulator of the cytoskeleton, this NWAS protein, so they can no longer respond to the bacterial signals and push up their cytoskeleton to form this pedestal that the bacteria perch on and release their toxin and cause diarrhea. So that's all shown really nicely in their cartoon in figure 4J. So they conclude by saying that we've shown that DRD2 is actually mediating host microorganism interactions in the gut, and it's another form of colonization resistance. They do point out that the amount of tryptophan they had to provide was fairly high, because if you're starting to think, okay, should I just have a large dose of tryptophan if I've eaten a pink hamburger. But they do say it is a fairly specific effect on these cells, so there's potential that they could follow up on this. Somebody could use this as to guide development of prophylactics or therapeutic agents. But in any case, it gives us another insight to the magnificent innate immune response of our body to protect us against these pathogens that can cause these pedestals to form in our gut, and does give the field a chance to think about how we could interrupt this prophylactically.
Turkey.
Right.
Eat more turkey.
I know.
Well, I was just going to say that. I think every American who's ever seen that episode of Seinfeld where George Costanza, falls asleep because he's eaten too much turkey, that's probably the only instance where the average American understands the effects of tryptophan. But
I mean, it's really interesting because the dopamine receptor, right, that we have this not only from this perspective, like, can we treat the symptoms? Because it's sort of not, in a way, these infections move so fast, like how much are antibiotics going to be helpful? But can we treat the symptoms, right? Which is always hard. But also this sort of gut-brain connection is what I'm thinking about here with the dopamine receptor. Because we're always like, oh, what receptors are you using? It's probably something boring, right? But that it's dopamine is really, I think, also really
cool.
It's
Tell us about this investigator.
This investigator, I think, is great.
Wow.
Samantha sent a really sort of about where she sort of started and why she's excited about it. Like many of us, she really got excited about microbiology as an undergraduate at the University of Scranton in biochemistry, cell and molecular biology. They have a seminar during their freshman year that they can take where professors sort of, I guess, talk about their research. And she was really enamored of Dr. Michael Solzinski's group and work. And she worked in his lab actually for the next 3 years on Burkholderia gladioli. But she also received, and these are really great fellowships, any undergraduates out there, these undergraduate research fellowship from ASM, which basically allows undergraduates the chance to present their work at the ASM general meeting, which is amazing and huge and has everything you could possibly want under the sun in microbiology. But also these students get together. And so that's also really nice. It's a really nice fellowship, summer fellowship. But when she went to that meeting, she heard Dr. Laura Hooper give the keynote and talk about the gut microbiome. And she'd never really heard about that at that point. I mean, she's a college junior, but she remembered thinking it was cool. And so when she went to graduate school at Cornell, she sort of sought out Dr. Chang's lab because they work on the gut microbiome. And so that's kind of how she started. And she wants to also make sure not to thank only Dr. Chang, but also Dr. Jingjing Fu, who did the metabolomics. I think her most exciting part, and I love this because I try to get my students to do this, was she had a hypothesis on her PowerPoint presentations that she'd used for this project that about all the proteins downstream of DDR2 signaling. And when she actually inhibited the pathway, being able to check off that slide and say, my hypothesis was correct was really exciting.
Made her feel like a genius, I'm
Exactly.
sure.
I'm a genius. I have a hypothesis. But if you really think through your hypotheses, you too can sometimes become that genius.
Yeah. But we should just tell the listener, usually we call it research because it doesn't usually happen on the first try.
Usually you have like 5 hypotheses. It could be this, or this.
Yep.
And then in terms of advice, her biggest takeaway is perseverance is key. There's always challenges, there's setbacks. Sometimes she felt out of place, sort of imposter syndrome, which I think many of us feel at different stages of our career. But really, I think just focusing on the work seems to have been, for her, the solution, and just working through the setbacks as they come, not kind of getting overwhelmed by them. So perseverance or resilience, I think, is her message. So I really like that she got inspired as an undergrad and kind of kept doing microbiology.
I remember that keynote. I think, Michael, you were with me.
Yes, it was fascinating.
Sitting next to me.
10 years ago.
Yeah, I remember that far back sometimes. Thank you, Petra. Thank you, Michele.
My pleasure.
That is TWiM 307. Show notes, microbe.tv/TWiM. If you enjoy these programs, we'd love to have your financial support. You can go to microbe.tv/contribute. And questions and comments, TWiM@micro.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Michael Schmidt, Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levin's at Washington University in St. Louis. Thanks, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.